All right. I want to see more of you than that. You know how lonely I get in my kitchen. Okay. All right, today's lab begins on page 39, and we're doing the chemical composition of foods. And so I will switch and I'll show you what you need to get out of your lab kit and what you need to have from your kitchen. All right, so I grabbed a couple paper towels. You may want a paper towel or napkin to put stuff on, but you'll need today your goggles and your gloves. So grab those. And today, let me put this over where you can see it. You'll need your labeling tape and your marker, whatever you use, and you will have to have your tubes labeled today. Okay, so find something to label your tubes. And you're going to need 5 test tubes. I have 6, but you will need 5 test tubes. And your test tube rack, obviously. I have my plastic cup, and in that I have my glucose and my starch from last week. So my glucose and starch solutions that I have left. I think you have 2 transfer pipettes. You want both of those out. I have my ruler. We may or may not need that, but you may want to pull that out. The same with your cork borer and your applicator stick. You may want to have those out just in case you want to use them.
I don't have any more glucose and starch.
Say what?
I don't have any more on my tubes.
Then what I would suggest is add just a very little bit of water, like 1 ml.
On both?
Bottoms of them, because you're going to need some for today. So hopefully you'll have enough in there to be able to get a little bit of reaction out of. Okay, so just add just like a 1 ml up to the 1 ml line. Or I guess it's up to the 2 ml line you'll have to do, add a little bit of water. Hopefully that will work. Okay. Also, you'll need the rest of your glucose test strips. And you'll need your protein test strips. They look identical. And while you're in your bag, get out your last vial of iodine. And you also want your brown paper bag squares. Now, other things that I have here, I have my graduated cylinder and I filled it up with water. It doesn't matter how high, I'm just going to use it to use water out of, and I don't need that much water. But I'm using my beaker and right now it's holding an egg. You do not have an egg. You don't need an egg, so don't worry about that. This is the egg actually prepared or mostly prepared, so you don't need that either. And then I have my spoon that I'm going to use to mush my samples. I have a knife to cut if I need to make something smaller. And I have my potato. My potato is a purple potato. So you'll need your potato, your food source, whatever that is. I have a variety here. And you need oil of some sort. You only need a drop of oil. So I've got my toasted sesame oil, actually, that's at the bottom, and it's old, so I'm getting rid of that. Another thing you can use instead of oil is if you have something like this, Pam, or a spray, you can try that as well and see how that works. Okay, everybody with me with all that stuff? Everybody got theirs out? All right.
Do we need the scale?
No, you don't need the scale today. Next week. Okay, so I'm going to start out by introducing the lab just a little bit. So I mentioned that it starts on page 39, and you're actually lucky compared to Tuesday's lab, because remember now we have been over carbohydrates, right? So you're a little more familiar with some of this than they were. But starting on page 39, you probably recognize in the diagram on the bottom of the page, dehydration, synthesis, right? Same thing as condensation, and hydrolysis. Those opposite reactions, one building up molecules and one breaking down molecules. The molecules that we're going to use today are all based on that mechanism, being able to build it up and break it down. Okay, so that's why that's covered there, but you're familiar with that reaction. And you'll see under each of these different organic molecules that we're going to cover, you'll see that it will describe how they go together. And you'll see that each one of them is that dehydration synthesis reaction. But we are limited at home. And we're limited at home because we don't have some of these reagents. So we've kind of adapted. Part of that adaptation is those dipsticks. So the reaction that you're going to see in here is not going to be the same one that you're actually going to perform. So we're not going to spend a lot of time going through what's written in here. So I just want to point out that we are going to test proteins. So I'm on page 40. And we're going to test proteins, and we're going to use a different method. We're going to use our dipsticks. So the color reaction that you would see in there is not what we are going to see or do. And page 41, at the bottom of page 41 is where they start carbohydrates. Now, you probably remember from yesterday that glucose is a carbohydrate, and it's a monosaccharide, right? That's a monosaccharide. If we attach a bunch of glucose molecules together, we get a polysaccharide. And there were 3 of them we talked about yesterday, but you're familiar with starch because we've actually tested for starch, haven't we? And how did we test for starch? What did we add to the starch to get a reaction?
Iodine.
Iodine, absolutely. We added iodine, and hang on just one second. For some reason, my volume is not very good. So let me see if I can turn that up a little bit. Okay, so we added iodine to starch and we got a dark color. We saw in potatoes in our microscope lab as oval black bodies, right? Everybody remember that? So we're going to be using that reaction today. We're going to look for starch using iodine. But then on page 44, we're actually going to look for glucose using our dipstick. Okay, so we're doing 2 carbohydrates. We're doing our starch, which is a polysaccharide, and we're doing glucose, which is a monosaccharide. And then as we move along in the chapter on page 45, we're also going to test for lipids. Now, the lipid test we're going to do is very basic, very simple. And we'll talk about that when we get there, but we don't have a dipstick for that one. And what I'm going to ask you to do as we go along, so we're going to do 4 tests. Our 4 tests are going to be for protein, for starch, for glucose, and for lipids. Okay, so that's what we're going to do. Now, on each one of those tests that we went through, there is a table for you to put your results. What it doesn't have is your food source. So you can add your food source right to the bottom of that table. And we're also going to change some of the things that we're testing. Okay, so whereas I'm on page 41 and I'm looking at proteins, and my protein table says we're going to do distilled water, and we're going to do albumin, pepsin, and starch. Okay, we are not. We're only going to do 2 things. I'm going to provide a third one for you. Okay, but we only have 2 protein dipsticks. So we will change what we're going to be doing in that particular table. Okay, but then you can add your food to the bottom so you can record the result. All right, so that's how today is going to work. And I do want to go to the top of page 40, right at the beginning. And I want to talk about controls. So what we're going to do today, we're going to do a couple things. We're going to do a bunch of things, right? But I want you to pay close attention in each test to the controls. And what I mean by controls is, if I have a tube with water in it, pure water, would I expect to get a positive test for glucose on it? Should there be glucose in water? I should be seeing all these heads going, no. Water isn't sweet. We don't have carbohydrates in water. So if I were to get a positive result for glucose in my water, I have a problem, don't I? Right? I don't know what that problem is. First problem could be that my glucose test strip isn't working. The second one could be maybe I didn't test the water. Maybe I thought I did, but I tested the tube next to it. Could have been my mistake. Maybe there is some residual glucose in that tube. Maybe I didn't clean it really well, and so there's something in the bottom of the tube. So a negative control is going to tell us that our system, it's going to give us the first indicator that our system is working correctly. So in each of our tests, we're going to have a negative control. That negative control is going to be water, because we don't expect water to have glucose or starch or protein or lipids. Okay, so we're going to run a negative control on each one of these tests that we do. Now, also good practice is we need to have a positive control for each test. For one reason, it gives us something to look at so we know what a positive looks like, right? So we have a good indicator. But for the same reason that we do a negative test, we want to make sure our system is working, we do a positive test. So let's say I am going to do a glucose, I'm going to do my sample of glucose, and I'm going to do my dipstick on my sample of glucose. Do I expect that to be positive for glucose? Of course I do, right? Because it's glucose. So I expect that my test strip will read positive for glucose in my glucose tube. That's a positive control. If my dipstick does not read positive for glucose, once again, I have a problem. Either I didn't pour glucose into that tube, or my test strip is not working properly. Maybe I tested the wrong tube. I thought I tested this tube, but I didn't. I tested starch instead. Okay, so those are things that we run. We run a positive control and a negative control with each test that we do. Okay, the exception for us, sort of, is going to be the protein because you only have 2 dipsticks. Okay, but I'm going to take care of that problem for you. Okay, so seems like I was going to tell you one more thing about, oh, the other thing is we're going to work on hypotheses, right? We know what a hypothesis is, and next week, we are going to actually be doing that experiment where you're going to do the experiment and you're going to develop hypotheses and you are going to write a paper. Okay, I will give you all the guidance you need through that process next week, but we're going to get a little practice today thinking about hypotheses. So for each of these tests, what I want to do before we perform it is and you can write it right in your book, write it right near the test. And you want to write sort of an if-then statement. Not all hypotheses have to be in an if-then statement, but let's say, well, let's do it. Let's start and we'll do it then. Better idea, because then you'll have a better idea of what we're looking at. So get your goggles on, take your selfie, gloves, and I'm going to switch our view. And you guys, make sure to slow me down today if you feel like I'm going too quickly. Okay, if you can't keep up, it's not going to hurt us to slow down a little bit. I have a tendency to kind of get going, and since I've done this already a few times, let me know if you need to slow down. Okay, so the first test we're going to set up today is going to be our lipids. Now, picture yourself, you have gone to Five Guys and you've gotten that whole bag of French fries, right? And they're all in the bottom of the bag, and you've got your burger, and you drive it home, and you get home, and what has happened to the bag?
Got soggy.
It got soggy. That's right, because that is oil from those French fries, right? That has gotten into the bag, and the bag has a funny look then, doesn't it? Okay, it looks wet, but it doesn't really look wet, does it? It's kind of almost like a translucent look. So our first test is going to be using oil, okay? And so all I'm going to do right this minute is I'm going to label this, because then we're going to prepare our test tubes. So the first thing I'm going to do, remember, I need a positive control and a negative control. So I am going to have water as my negative control, and that's going to be my negative control for every test. And then I'm going to have oil, because that's my food that I'm testing, or not that that's not my food, that's my positive control. I'm going to put positive and I'll put negative. And then I'm going to have my food. And I need to get all these prepped. But you know, thinking about this, you know what that bag looks like when it gets greasy. So this is not a very testable hypothesis because we don't have a way to say exactly what it looks like. But make a hypothesis now. Think to yourself, if my food has oil or fat in it, then what is your result going to look like? Okay. And in the process,
Sorry, excuse me.
yeah.
So it's water, oil, and fat
Your food.
food.
Your food is going to be the third one. Okay, so think about your hypothesis. First of all, do you think your food will have oil or fat in it? And if it does, what will the bag look like? So I'm going to make popcorn, and if I say, I believe that popcorn has oil in it, and if it does, my little bag square will look, whatever your description of that look is. I'm going to say translucent. Okay. So go ahead and do one for your food, and we'll get all this set up.
Professor, can you explain that again?
What am I explaining? The hypothesis?
Yeah, what are we doing with the brown bags?
Okay, don't worry about the brown bags yet. Worry about the hypothesis that you're supposed to be writing. Have you written your hypothesis? What do you think, Carrie, what do you think your food, what's your food?
I have crackers.
Okay, do you think crackers have oil in them?
Yeah.
Okay,
Maybe.
all right. So if crackers have oil in them, what is your result going to look like when you do the lipid test? What is a positive result? Think Five Guys. What's that bag going to look like if you've got oil in it? If you've got oil in your food? You see what I mean? Okay, do that, and then we'll go over this because we haven't started this part yet. We're just getting set up.
Do we need to write it down or just think about
Yeah, write it down right in your book somewhere.
it? Okay.
The lipids are, and the table for lipids are on the top of page 46. So if you want to write it right next to that, you can. Vashta, did you get those labels? Okay. Okay. So let's make all of our test tubes. So the first thing, I've already got labels on mine, and you should have 5 labels for your test tube. The first one is going to be your food. And you can just write food if you want to, or you can put cracker or whatever you have. So the first one's going to be food. You'll have one that says water. And glucose. And starch. And I'll leave these so you can see them. And potato. All right, so there they are. So you want those labels, and you want one on each test tube. Okay, so there's mine for food and water. Okay. If you can't read those, let me know. They're kind of bleeding on my, my marker's bleeding on my paper towel there, so. Okay, are we good with our labels? Anybody need another minute? Okay. Okay, so what we're gonna do now is we're gonna fill these test tubes, and we don't need much of anything, but let's start with water. And so I'm gonna use one of my transfer pipettes. Let's see if I can put this up a little higher and see a little bit more. But I'm going to use one of my transfer pipettes. And remember, I have water in my graduated cylinder. And all I'm going to do is add 2 milliliters. And remember, the milliliter line is right below the bulb. So I want to add 2 milliliters to my tube that's labeled water. So I have about that much water in the bottom of my water tube. Not very exciting, certainly. Remember, that's my negative control. Okay, my next test tube is mine is glucose. So find your glucose tube. And that's where you want to mix up your glucose from last week. And if you have 2 mils or less, pour it in. If you have more than that, estimate about how much is 2 mils, just like your glucose. It doesn't have to be perfect. So all I'm going to do is put it up to my water line. Okay. So about where my water line is, I'm going to fill up to glucose. If I don't have quite enough, that does not matter. Okay. Fill up what you've got if you don't have quite enough to get to where your water line is. And I am mixing my starch. Remember how your starch will settle on the bottom. So mix your starch. And you can do the same thing in your starch tube. So your tube labeled starch, you want about the same amount as you have in the other 2 tubes. So right now I have water, glucose, and starch in the bottoms of each of these tubes, and you can see they're about the same height. Now we've done the easy ones. Now we have to do the ones that are a little more difficult. So first of all, let's take our potato. And you want, in this case, you may use your cork borer and applicator stick if you want to, but you don't have to. All you really need is a tiny little piece of potato. Think the little 1 centimeter piece that we used for our test tubes last time when we did the hydrogen peroxide and the bubbling. So you really don't need very much. So I've got a piece this big. Okay, that should be plenty. And what I want to do with this is I want to mush it just like I mushed it last time. Now I'm going to use my knife to cut it up and then try to mush it a little bit more and see how well I do. See if that helps. But I want to expose the cells in there just like I did before when I mushed up the potato for the catalase. I want what's in those cells. So I'm going to mush up this as well as I can with the back of my spoon. And once you've done that, kind of collect it like you did last time and put it in your tube labeled potato. Mine doesn't want to go down. No, there it went. All right, it went down all right. All right, but now I need to add some water. So I want about as much water as I have in the other tubes. Now, you guys who have things like crackers, you may find you're gonna have, oh, well, we're not there yet. Never mind, forgot I said anything yet. Okay. So now I'm gonna mix my potato in that water, just like I did last week when I mushed my potato in water. My potato is purple, so have you ever had a purple potato? I think they're pretty good, but my family was not convinced, which is why I have purple potatoes left.
I like them.
I do too. I don't know why they don't like them, but they didn't, I shouldn't say they didn't like them. They didn't like them as well as just the regular yellow potatoes or russets or whatever other ones we've had, so. But I was all right. Okay, so if your potatoes all done and mixed, you can leave that test tube. Now the last one is going to be your food. And before we do the food, we're going to set up our lipid test. Okay, so the first thing we want to do is go back to our transfer pipette and we're going to put 1 drop of water on our piece of paper here. Okay, so I'm going to put 1 drop of water. And that's not very impressive. I'm going to try rubbing it around a little bit. Okay. So it's soaked in right now. That's water. Make sure you keep your transfer pipette that had water in it separate from the one that you're going to use for oil, right? You don't want to reuse your oil one. Now, there's a couple ways to do oil. If you chose something like this, the spray, just make sure you don't spray everything. Just make sure you only spray your one piece of bag here. I'll tell you my secret with this worked pretty well, is I only need a bit. So I kind of tipped it over before I started and got the top oily. And then I was just able to pour a little drop from the top. Whoop, I said that and it worked yet better yesterday than today. But I was able to get a little bit from the top, and that way I didn't even have to use my transfer pipette. But if you have to use your transfer pipette, just make sure then you put it by your sink or in your to be cleaned pile so you don't reuse it. So you've got a drop of water and you've got a drop of oil. And now the way we're gonna make our food for this is, well, I don't know about you, but I've gotta wash my spoon because I don't want potato on it. And I'm gonna do the same thing with my knife so I can cut my food. So hang on a sec. If I were to carry over potato to my food sample, what might I actually have a positive test for that may be erroneous or wrong?
Oilers.
Starch.
A starch, right? I might end up with a positive starch test, and I don't want to do that. So I'm going to, I think I'm going to do my popcorn again. It looks a little sad today. But I'm going to try to crush up my piece of popcorn. Now, what you need, see how big my popcorn is and how big your potato was? That's about the only size you need for your food. Okay, so do very little of your food. And I'm going to try cutting my popcorn first. Yesterday, I found popcorn was a little challenging to deal with. Try not to cut my finger while I'm cutting a piece of popcorn. Okay, so I've got pieces here and now I'm going to crush them. So I think this is what I found yesterday too, is that they do not crush as well as you would think, but I was able to get results on it. So let's see how well I can crush this piece today. All right, so I've got some crushed pieces and cut pieces, and as I say, I really don't know whether I didn't do very well. Hopefully you had better luck. I think my fresh popcorn worked a little better. But that first thing that's going to happen is I'm going to put the big pieces of my food into my test tube, but save. Either if you crush something on the bottom of the spoon, take a little bit of that sample and put it, whoop, I just sent it sailing. Wipe it on the paper bag. Okay, so try kind of wiping it from the back of the spoon, or in my case, I took a little piece of popcorn and I'm rubbing it right on the paper bag. Okay, so hopefully I'll be able to see that oily residue on the paper bag. So you're taking your crushed food and you're wiping some of the food or the back of your spoon on your paper bag. And the rest of that mush stuff is going into the test tube that says food, or maybe you labeled it.
You want to split water with this?
I do, but while you are, once you've got your food on your piece of paper bag, go ahead and move those right out of the way. We're going to come back to those later. Okay, and you want to add about the same amount of water again to your test tube. So all of your tubes have about the same amount in them. Don't sweat it if they don't, okay? If one has a little bit more or a little less, that's fine. And just make sure to mix your food in well with the water in the test tube. I can't even get my popcorn down to the bottom. Okay, but I'm gonna mix it, and I can start to see that the water that I have in the test tube is starting to be kind of gunky, a little more cloudy. So I know that despite the fact that I didn't crush it very well, it is mixing with the water, so. Now, this is where I was mentioning, those of you who have crackers, sometimes the cracker sucks up all the water. Anybody had that happen? So sort of like using bread and you end up with not much fluid and the crackers stuck in the bottom. If that happens to you, you are probably going to have to add more water. So you may have more water with your food, just in order to make sure that it's not all stuck in the bottom and solidified. Okay, so the last one I'm going to do here is the egg. And what I've done is I've broken an egg and I've removed the yolk. So this is just the white of the egg. And probably you know that the white of egg has protein, right? You've probably heard of it as albumin. Albumin is a protein, and it's a protein that we have in our blood. We have a lot of albumin. So I'm just going to take.
We don't need an egg, right?
No, you don't need an egg. I'm just going to take 1 ml of the albumin, and I'm going to add a 2nd ml of water just to mix it in. And I'm just going to make a solution with this egg. All right. So I have my 6. You should have 5 test tubes all ready to go. You guys good? Okay, the first test we're going to do will be on page, I say on page 41, but you're going to use the table on 41. And we're going to do proteins. And we know all about proteins. We talked about proteins. We know about enzymes now. So we are looking in our food source for proteins. So what we're going to do is we're going to do all 5. Oh, no, we aren't, because we've only got 2 test strips. I lied. So you are going to do distilled water. Okay, you're going to do the negative control, and you are going to do your food source. That's all you're going to do. And I'll show you how you're going to do that. But here is your protein test strips. Make sure you pick out the right ones. And they look just like the glucose. So you got to be careful that you're using the correct ones. So I'm going to do the positive control. I'm actually going to do albumin, or the egg. You're going to do water and your food. So I'm going to take the egg first, and I'm just going to quickly mix it up. And what I'm going to do to test this is I'm going to hold my test tube on its side. So some of the water or the solution comes down the test tube. And I want to make sure that it makes contact with my dipstick. Now, this test takes 60 seconds. I'm just going to dab that to make sure there's no extra stuff on it. And I'm going to let it sit for 60 seconds. So you want to do that with the water. And then you want to do that with your food. So I'm going to do my food now. We'll see how well that goes. Okay. So I'm just going to set my 2 there.
So just our food and then our water?
Yep. All right. Basically, what I'm doing is a positive control for you. This wouldn't be the way we would normally do it, but with our limitations on test strips, that's what we're going to do. So I want you to look, and here is my protein. Okay, negative is down here. I have, looks like over 10 grams per liter of protein in the egg. That's the positive control. So that is done. And now I'm going to examine my food using this same color chart. And my food is negative for protein. Okay, you want to read your food and your water tube. Ah, we forgot to do our hypothesis on that one. Okay, I have not been keeping up with the chat. It doesn't matter if you're using a potato chip, Pam. And does it matter if you test positive for starch? We haven't done starch yet. And we'll examine the oil when we get to it. It doesn't matter, it's what your results are. Okay, so there's not a positive or negative. I mean, there's not a particular result we're looking for. Okay, did everybody's water come up negative? Good.
Yes.
So our negative control is working. How about your foods?
Just my food is 0.3.
0.3. What was your food again? I can't remember.
A cracker.
Oh, a cracker. Okay, so it has a little bit of protein in it. Some of these you can actually look at the nutrition label too when you're done and see how it compares to what you got.
Mine
Hi.
has protein.
Whoop, go ahead, somebody. Bailey, what?
Mine has protein in it.
Yours has protein, and what was yours?
The cheese.
The cheese. Oh, that's not a surprise, is it?
Nope.
No.
Mine's like in between the negative and the 0.3.
Okay, so it may have a touch of protein in there, but what is yours, Haley?
A banana.
A banana. That's interesting. What we end up doing in class, because there's a lot of stuff, when we do it in lab at school, we end up doing a lot of looking up things on Google to see if they should have had protein or whatever. So you can look up after and see whether they do or not.
On the label, it says it has 3 grams.
The banana?
The cracker.
Oh, the cracker. Yep. So it does have some. Yep. Remember that when it says it has 3 grams too, it's doing by a certain amount. And we're doing much less usually than their serving size. So we would expect that we wouldn't see as much. Anybody else have a positive on theirs? Gabe, what did yours look like?
I was using crackers and it looks like it had 1. So like 1-ish.
Okay. It wouldn't be a surprise, I don't think, if some crackers had protein. I'm trying to think. But different types, we know there's all sorts of different types of crackers too. What about you, Dominique? How'd yours look? Are you there, Dominique? Is your protein positive? This is why I like visuals. Okay, So let's move on. You can discard your test strips, put them right in the garbage. Get those out of the way, and even get the container right out of the way so you don't mess that up.
Do you want us to keep taking pictures?
Oh, good idea. Good idea. Take pictures of everything that you can take pictures of. You never know when they might come in handy.
Bueno, bueno.
Okay, the next test.
Do you want us to throw this out too? The little bag?
You don't throw it out, put it back in your kit, but just get it out of the way. All right, they can repackage those and reuse those if we have to do online again. Okay. Next is on page 44, we are going to do glucose. And we're going to do it because it's a dipstick. So let's get that done. And in this case, we have, count your dipsticks, dump them out and see how many dipsticks you have. Make sure you have 5.
Mhm.
1, 2, 3, 4. Yeah, I have. You have 5 or 6 maybe? Okay, so you have enough to do all of these. So what I want you to do is I want you to go down through and you're going to do water as our negative control, glucose as our positive control, and then we're going to do starch, potato, and our food as well. Okay, so go on down the line and do each one of those. Make sure that you keep track of where they come from, right?
You said glucose was positive.
Glucose is a positive control for a glucose test.
Oh, okay.
So I'm just going to go through and dipstick on mine. And then remember, it might take up to 3 minutes.
So we testing the water right now?
You're testing everything. Everything you have a test tube for.
Okay.
And once you get done, write your hypothesis while you're waiting. Write your hypothesis. Do you think that your food will have glucose? And if it does, what will the result look like on your test strip? And if you think it doesn't, what will it look like? So give me a color reaction. Or what I did yesterday when I did this one was I said, I think that my popcorn will have glucose, and I will know that because my glucose test strip will read 100 or above on the color chart. Okay? Or I could say it's going to be yellow, or it will read a shade of green on the color chart, depending on what my hypothesis is.
We haven't done table 4.4, right?
I don't know.
On page
No.
43.
No. Anybody want to tell me what your hypothesis is? Nobody wants to tell me what your hypothesis is? Make it easy. Tell me whether you think your food's going to have glucose or not.
I think my food and potato is gonna have, but the other ones, the water, it's just because I don't have I didn't have the solution, so I was just water that I pour on the glucose and starch.
Okay, that makes sense then.
So, but yeah,
Megan, what is your food? Do you think your food will be positive or negative?
I think it will be maybe positive.
Okay, so Megan thinks her food will be positive, so her hypothesis probably looks something like, I think I can't remember what your food is, Megan. Will you use strawberry?
Bread.
Okay, bread. Ooh. I think my bread will be positive for glucose. And therefore, my test strip will show 100 or more for glucose, something like that, right?
Yeah.
I'm sure that's exactly what it was. All right, Darius, is yours going to be positive or negative? What do you think? I couldn't hear you, Darius. I know you took off your mic though, but I don't know. You can put it in the chat. Positive or negative, do you think, for your food? I don't think you can, I can't, I know. All right, you say negative. And all right, so let's look at our results and see how they look. First of all, do your negative control. Which one's your negative control?
0.
Water.
Our water should be
Yeah,
our
water.
negative.
Sorry.
That's okay. Our water should be our negative control, so make sure that that is negative. And then look at your positive control, which is glucose, and hopefully, if you didn't have the same problem as you carry, hopefully you get a nice reaction in the glucose because that should be positive. And then you can move along, and starch, remember, is a polysaccharide made up of a whole bunch of glucose molecules. But if starch is in the form of starch, do we see any glucose? Anybody? I don't see any.
No.
Isn't that interesting though? Because it's made up of glucose and yet we don't see a reaction for glucose. That's pretty cool. I'm looking at my potato. How does your potato look?
Positive.
It's positive. Mine is really positive. And I didn't know if mine was really positive because it's old and been sitting in my refrigerator for quite some time, or if it's the type of potato, but Bailey got positive. How does everybody else's potato look?
Mine's positive.
Mine's positive.
Is anybody's potato negative? Huh, okay. So we know that probably some starch is breaking down in there, right? Okay, so let's go to our food. And what is your food doing? You've now tested your hypothesis. What is the answer?
Mine was positive, the cheese.
Positive.
Well, did it
Positive.
match your hypothesis? Mine didn't, right? I said I thought mine would be positive. It is okay to have your hypothesis be wrong. When you write a paper in science, your hypothesis is the best guess using what you know, and then you do the testing. So the hypothesis comes first. That means that not quite as probably as often as not, but you're going to very frequently have your hypothesis wrong. In science, basically, we say my hypothesis was not supported by my research or my data. Okay, that's what you do. You don't lose points. That's not how it works. It just means that you were not able to prove your hypothesis or support your hypothesis. So mine is wrong, and that's that. So when you do your paper next week, when you're writing it up, you're going to make those hypotheses ahead of time. And in your discussion, you're going to look at the results that you got by doing your research and determine whether your hypotheses were supported by your data. Okay? And we'll talk all about that next week. But that's why we spend so much time with hypotheses. All right, once you've read these and taken pictures of them, you can discard the dipsticks. And then what you want to do after you've discarded the dipsticks is get rid of your glucose. Don't get rid of it. Put your baggie back in your test kit. You're done with that. I'm going to put my brown paper square baggie back in there too. Get rid of my popcorn pieces that are all over the place here. Okay. So next and the last test is going to be for starch. Okay, so our polysaccharide, another member of our carbohydrate group. So in order to do that, I'm going to need my iodine. And what I want to do before I do this chart is on the top of page 43. And you are going to be able to do each one of your tubes. You should have enough in your starch container. So you're going to test your water as your negative control, your starch as your positive control, and then glucose and your potato and your food for this. So before we do that, write your hypothesis for your food. Okay, so recall when you use the iodine that all you do, point it away from yourself and press the middle, you'll hear the pop. Okay, crush that a couple times. And then remember that this dropper tip is already open now. And what I want you to do is start and add 1 drop of iodine to the water. You may need to shake it down. Whoop. Well, mine got stuck on the side. Let's see if I can. Well, there we go. Got that one in the bottom. Okay, so mine looks a little like that. Now, should water have starch in it? No. So this gives me an idea of what I would anticipate for a negative result. That would be iodine and the regular color that would occur for a negative. So that gives me something to compare to. My starch, 1 drop in my starch. Okay. And yours may be darker than mine. I used an old starch, but it's still, let's see, Bailey, hold that one up again. Oh my goodness, yeah. Is that what yours looks like? Yeah, yours are all a little stronger than mine. I watered mine down a little bit, but you get the idea. So that is a positive result. So
Professor,
now-
I
Yeah.
thought I washed my starch too, but look, it turned green, and I thought it was water.
So it turned green, or is it, so this is water with iodine?
Yeah, I know, but my starch, like I didn't have starch, so I poured water, but it turned green, and the water that I had here is like
Okay.
regular.
Okay, so obviously I would say this is a perfect example, Yokari. That's why we do a positive and negative control, right? So, but I'm guessing since you didn't have starch in there, that there's probably something else in there. So you're just gonna have to go with it because we can't retest it, right? Because you don't have any starch.
No, I pour it on the starch too, right?
Right,
And then I pour it on the water, right?
right,
know.
but you put starch,
I think it had like a little bit of starch, probably like on the tube.
okay.
Yeah. Well, I don't know.
All right, so we've done the starch, we've done 1 drop of each. Go ahead and do the glucose, 1 drop of glucose. 1 drop of iodine into glucose, sorry, didn't say that very well. Mine looks awfully similar to the water. And then go ahead and do a drop in your potato. And a drop in your own food.
Oh my God. Oh, I'm sorry.
There's my food. That's my popcorn. My potato's a little harder to read, and I'm actually going to add another drop. If you find now you've got iodine in everything. So if you have a little bit left and you need to add it to a tube to be sure, like my potato, I'm going to add it to this tube because I'm going to add another drop and see if it gives me a better indicator. Because that one's pretty hard. The potato on this one is pretty hard. But I'd say my potato is positive, which makes sense. Your potato should be positive, right?
My potato is pretty easy to read.
Yeah. Well, remember my potato's purple. That's, I think my bigger problem isn't the starch, it's the purple color that it's got. but it did, it's changed. Well, you can't, it's changed pretty well. But this is my food. So my food is definitely positive for starch. Oh, that's interesting, Vashta. It's kind of a weird color, isn't it? See if you can add more iodine to it and see if you can get it to commit a little more.
My food was definitely negative.
Negative, huh?
It has a very weird yellow to it.
It has what?
A very weird yellow.
Yeah. Well, and the yellow is probably, if you consider that that is water, right? The iodine is going to stain it somewhat. Pretty cool though, isn't it? I was glad mine had something. All right, so make sure you take pictures. This is what I want pictures of for upload. So make sure that you take a picture of your starch tubes with your so you can see the results, and make sure that the labels are visible. So make sure the labels are looking toward me. And in case anybody was interested, I did just add, I had a little bit of iodine left, and so I added it to the egg mixture, and that is pretty unexciting. So egg does not have starch, or egg white, I should say, doesn't have starch. I think since I'm going to take a picture of mine too, so hang on a sec. All right, so I've got mine done. And I want to go back now. We can actually take your iodine container and throw that right away. You're done with that. Get that out of here. You can save the baggie and put it back in your test kit. Any baggies, things like that, they can reuse if they need to. So I don't know whether they'll choose to. And I'm going to look at my lipids, my oils. Now it's pretty easy. My water, because I spread it around, has completely dried. And there is no indicator left that there was anything even on it, little wrinkled, right? But my paper hasn't changed. So that's what I would anticipate as a negative result. My oil, however, you can see it best on here, has that translucent look. If you lift it up, you can actually kind of feel like you're seeing light through it. Okay. And that's what I would anticipate for a positive. And then I can look at my food. Now my food is a little oily. It didn't go through like the oil did, but it's definitely a different look on my bag. So now you can evaluate that hypothesis that you made. Now, I want to point out that that is not a good testable hypothesis, is it? It's kind of arbitrary. I think I gave as an example in class, I think I gave the sky is blue, right? The sky is blue as not being a very testable hypothesis. And that's what we have here. So this test would not be a really great test. And if you actually read the chapter or read in this lab, you'll see that there was another type of test that was less arbitrary. But this gives us an idea. Okay, so you can just be careful about throwing stuff down your sink. So in tubes that have food or potato, you may want to dump those in the garbage first. Otherwise, make sure that everything gets cleaned today before it goes back in your kit. We are done with the test tubes, so wash them real well. Make sure the labels are off of them. Obviously, anything that you've used, you want to make sure to wash. And if you used oil in your transfer pipette, my suggestion is to take your beaker, put some soapy water in it, and suck water up and down in your transfer pipettes, right? And then shake it. You can actually suck it up and shake it a little bit. That'll clean the whole thing, and just rinse it then with clean water, okay? So make sure that you get the oil out of your transfer pipettes or transfer pipette. Make sure everything's clean. Okay, these glucose and starch, make sure the tops are on tightly, and you can put those back in your kit. We'll send those back. Trying to think, anything else, obviously, you can put in your to be clean pile. But I want to go over one other thing before we leave today. So you can discard gloves, get those styling goggles off. Check out your lines on your nose. And we've talked, we have talked a bit now about doing a hypothesis. So, well, actually, I apologize. I'm going to do this first. So on page 49 are the summary questions. And going down the summary questions, you don't need to do number 7, number 11, 12, or 13. And number 17, I want you to change the wording a little bit. Everybody
What
with me on 17?
What page is this on again? I'm sorry.
49. So you can completely skip 7, 11, 12, and 13. And on 17, it says, if a sample of water you test with, and I want you to change Biuret to iodine, and then change turns, instead of purple, turns black. Okay, so you're just changing the view of the reaction to something we actually did. Okay, so it would say something like, if a sample of water you test with iodine turns black, what should you conclude about the results of your experiment? Okay, so you're going to submit those for your summary questions. And then I want you to submit for your photo this week, the starch. Okay, make sure that, obviously, I want to see labels with the starch, but I want to see your starch results. All right, and now I am going to, I think, I'll be right back. Let me make sure I'm signed in here so I can share my screen. I'm not taking anything for granted about my computer hitch hookups today or whatever. It's been giving me such a problem. All right. All right, I'm going to share my screen with you and I'm going into the lab. Okay, so we are on lab 4. That's where your uploads are going to be. But this lab, we are not having a quiz for. Don't get all excited because you have a pre-lab assignment for next week. Okay, but when you come to lab next week, there will be no quiz. But you have to have submitted your pre-lab assignment. So your pre-lab assignment is going to be doing hypotheses for next week's lab. So I want to go through that a little bit. I'm going into lab 4. Can you guys see that all right? You saw me go into lab 4, it's following it? Okay.
Sorry,
So
are we done with the experiments?
say that again, you Carrie? What?
Are we done using the lab kit?
Yes.
Okay.
Okay, so here is the summary questions that are due, the photo upload of the starch tubes. That's where you're going to submit those. But here is the pre-lab for lab 5. And there is a fillable form. And I'm going to take you into that. But first of all, let's recall from last week the terms hypotonic, isotonic, and hypertonic. Okay, so I want you in the right frame of mind. This gives you instructions. What you should do before you fill this out is to read, starting on, the lab is short. I don't mean short lengthwise, but to read. Let me find it. The lab for next week starts on page 50. And it tells you what you are going to be doing. And you want to read that and then fill out your hypotheses. So what's going to happen is you're going to have 3 different hypotheses. One is going to be which solutions that we're using are going to be hypotonic, which solution or point is going to be isotonic, and which is hypertonic. Okay, so let's think of the terms first. Let's start with osmosis. Osmosis is movement of water through a permeable membrane, right? A semi-permeable membrane. What we're going to be using is a potato cell. Okay, so we're using potato cells. And we're going to put the potato cell in, if you're on page 50, look at the table on the bottom, and we are going to put potato cylinders like we made for last week's lab. We're going to put them in 0% sucrose, which is water, 3.5% sucrose solution, 7% sucrose solution, 10% and 14% sucrose solution. That means that as somewhere along those 5 different solutions, we should reach what's called an isotonic point. That's the point at which the solute concentration on the inside of the cell matches the the solute concentration on the outside of the cell. Okay, does that sound familiar? I'll make sure to go over this in class tomorrow to review those terms for you. But you're going to have 5 solutions. You're going to have cylinders of potato in each solution. And you want to determine which solution or solutions are hypotonic and hypertonic to your potato cell, and what point or which solution is isotonic. Okay, so that is going to be what you write for your hypotheses. So I'm going to move this into this assignment, or I'm going to pull up this assignment. Let me see where it went. There we go. Whoop. Okay. So this is the pre-lab assignment that you have to submit before coming to lab. And it's a fillable form. And there's a couple questions on the top. First would be, will the cells in a hypotonic solution gain water or lose water? And what I want you to think about, if you're having trouble with that thought right now, remember when we took a potato cylinder and we put it in water, which is a hypotonic solution, what happened? You should have a picture of that. So if you aren't remembering, you should have a picture of that. Go back and look at your picture. And that should tell you whether the hypotonic solution, the cells gained water or lost water. Now, this is kind of in the wrong order, and I apologize, but remember that we want to make our hypothesis testable. So if you have read the directions, you will know how we are going to make our hypothesis a testable hypothesis. How are we going to evaluate that hypothesis? And that's going to relate to what we're going to do to the potato after it has soaked in the solution. Now, when we looked at it, when we looked at the potato that came out of water and came out of 10% salt, or that salt solution, we, for the most part, just looked at it, right? One was nice and rigid and the other was wimpy. But we did something else that made this testable. And that's what I'm looking for there. Not what it looked like, not what it felt like, but we did another step. All right, and then you're actually going to develop your 3 hypotheses. So I gave you a suggested form. If you want to use a different statement, you're welcome to, but this is the information that I am after when you write your hypotheses. So if it says, if whatever percent sucrose, and you know those percentages because those are in the lab, if X% sucrose solution is isotonic to the potato cells, then water will move in and out equally. The potato weight will not change, right? So for that one, all you really need to do is come up with what percent sucrose solution you think that is. Now, I'm going to make it even harder here. It doesn't have to be one of the solutions. You're going to be making a graph. So look with me on page, shouldn't have closed the book. Hang on, give me a sec. Look on page 52.
Wait, I just have a question. So we have to do the bottom on page 50 before class next Thursday.
No, no, you'll do that in class. I'm just showing you the solutions we're gonna use. And they're on that, they're written there. But you do need to read you'll need to read the lab, but you won't be able to fill it out until we do lab. So you're not filling anything out yet. Except
So
this.
for the pre-lab for lab 5, we don't need to upload anything, right? We just need to write it.
You have to write this page that you're seeing and you have to submit that. You have to upload that.
Which
before
page?
lab.
The
What?
one that you're showing right now?
I'm sorry, say that again?
Which page?
The page that I'm showing right now, the
Okay,
one that's in the assignment. It's in the assignment in this lab.
okay, thank you.
Okay, so on page 52, you will be making a table like this one. And if you notice, there is a point on, well, first of all, look at the y-axis and it says percent weight change. And at 0%, that means that no weight change occurred. So that would be our isotonic point, where it's 0. Now this data is arbitrary data. I just put stuff in so you could see how it works. So don't try to get anything off of this. But 0% weight change, if you follow that dark line all the way over, there is a point there that it matches up to, and the concentration on the x-axis is the percent sucrose concentration. So we are going to establish the points that you're seeing, right? And so for our hypothetical data, our isotonic point, because we're going to use a line, our isotonic point doesn't match up with any of those solutions that we had on page 50. So if you want your isotonic point to be 4.2, you can pick 4.2. Or you can pick a solution. You can say, I think my isotonic point is going to be my 3.5% sucrose solution. Okay, so you can pick anything. But remember, once you pick your isotonic point, remember that's where there's no weight change. One direction, so anything above that percent solution is going to lose weight. Anything that is hypertonic, meaning it's above that percent solution that you pick, is going to lose weight. So you're going to pick either a solution or you're going to pick a range of solutions, but that's going to be based on what you pick for your isotonic point. Your hypotonic point is going to be below your isotonic point. Okay? Does everybody understand that?
You said hypo is down, is below, right?
Hypo is below. That's going to be where your sample will gain weight. Okay? So you've got to put some thinking into this. And once you've kind of put the thinking into this, making your hypotheses should not be difficult. And if you use the lab and you go through the lab and go through the graph that you see, kind of put those pieces together, this should work out fine for you. Okay? But you got to make sure to do it, because when you come in next week, your hypotheses have to be ready. You don't wait till the end of an experiment to come up with hypotheses. It may be okay to get the wrong hypothesis, but it's not okay to start without the hypothesis. Okay? So you know where to find that. It is due at midnight. It'll be due midnight the night before lab. If you happen to forget, I prefer you didn't, but you can still get it handed in. Just make sure it's handed in before lab, okay? All right, let me stop sharing here. Okay. That is all I need to go through today. You're welcome to stay and ask questions or work on your assignments. Remember that the upload and the summary questions for lab for today's lab are going to be due on at midnight on Sunday. Okay, so I'm all done with you. So stay if you wish, or you are free to go. And have a really nice day. I'll see you all
Good for a few
tomorrow.
days.